Parkinson's disease 101. This is our first hybrid version of the 101 series. And we're really thrilled to have those of you who are joining us in person and all of you in Zoom. We have with us today Dr. Vikram or Vik Khurana. We're delighted to have him here. He's the chief of the Movement Disorders Division at Brigham and Women's Hospital. He's a principal faculty member at the Harvard Stem Cell Institute and an associate member of the Broad Institute. He's really interested in neurodegenerative disorders, including Parkinson's disease and related dementias, rarer conditions such as multiple system atrophy, MSA, and ataxias. He received his MD from the University of Sydney, and then he did a PhD here at Harvard in neurobiology, where he was a Fulbright scholar. He then did a neurology residency at Brigham and Women's and Mass General Hospital, a fellowship in movement disorders and ataxia at Mass General. And he did postdoc work at the Whitehead Institute at MIT. He did some really wonderful work looking at human stem cells, which were derived from patients with Parkinson's disease, looking at the underlying pathology and coming up with techniques to reverse some of that pathology in the patient-derived stem cells. This work has led to the discovery of novel drugs, which are in clinical trials. Recently, his lab has mapped the consequences of misfolding of alpha-synuclein, which is a protein that aggregates abnormally in disorders like Parkinson's disease. This mapping work sheds light on disease mechanisms and reveals novel functions for alpha-synuclein. The lab is interested in personalized medicine, in bringing stem cell technologies towards more targeted therapies. And on top of all of this, Dr. Khurana has founded 2 biotech companies that focus on the central nervous system and neurodegenerative diseases. So we're really happy to have him here today. And he'll speak for about an hour or so, and we'll have a lot of time, like 20 minutes for questions.
All right, I hope you can hear me. This is a great juggling act. Thank you so much, Farzad, and thank you to the Harvard Brain Science Initiative for initiating in-person meetings again. It's amazing. So I am balancing what's happening on Zoom with what's happening here in the room. So, so forgive me in advance if there are any technical issues, but we'll get started and see how we go. So Parkinson's disease 101, as the name suggests, this is a public lecture. Judging from the questions, there was a very broad range of audience members. I'm going to try and keep it fairly non-technical, but I'm very happy to answer more questions later. It's going to be a little whirlwind tour, mostly chronological. I'll try and really build a historical perspective on Parkinson's disease, and towards the end, maybe touch on some of the big questions that are keeping us from really preventing, slowing, or curing this disease. So let's get started. We're going to start and end really with our patients, which is as it should be. Now, take a look at this young woman. She's passed away now, but she is actually, she has contributed enormously to our understanding of this disease. And I'll recurrently come back to her story during this talk. But early in the 2000s, just at the age of 44, she was fired from her job for slowness, fired for slowness. At that time, no one knew what was going on, but she volunteered to engage in research. And as you'll see, it was very formative. Now, if you look at the pictures here, the images and the video, you can see that her hands are moving very slowly. There's a mask-like quality in her facial features. Her writing is slow, but also disorganized, a point we'll come back to later. The letters, the numbers are small. That's called micrographia. But the clock doesn't look quite right either. And we'll talk about that a little bit later. You get the sense that her movements are rigid. They're not smooth. And they're certainly very slow. This is called bradykinesia, slowness of movement. You can see that there. There's a loss of rate. There's also a loss of rhythm. She gets up okay, but it's still slow. The arm swing is lost on both sides in her case. The steps are small. When she turns en bloc, which means she takes a couple of extra steps to turn, and those arms are just not swinging, particularly on the left. She's a little unsteady, although she's holding her own. She needs to take a step back to stay in the same spot with a fairly minor pullback. That's called retropulsion. So this, of course, is Parkinsonism, and the cardinal features are slowness and rigidity, tremor classically at rest, although many patients don't have that feature, instability of balance, postural instability. And the most common cause of Parkinsonism, and I distinguish those terms, Parkinsonism is what we see in the clinic. Parkinson's disease is a pathologic diagnosis. The most common cause of Parkinsonism turns out to be Parkinson's disease, most commonly in the 50s and 60s. Some patients are younger. It affects 1 to 2% of people over 60, and it affects 10 million patients worldwide with a propensity for men. And just as in this case, this woman's case, 1/3 of patients with Parkinson's disease lose employment within a year after diagnosis. So despite, some very effective therapies, this is a disease that has tremendous impact on patients, their caregivers, and our society. So I'm going to take you through a whirlwind history. The first 2,500 years of Parkinsonism, really the era of dopamine, I'm going to call it. So actually, around 600 BC, a syndrome, it wasn't thought of as such, but a description of an illness of tremor was identified in old Sanskrit Ayurvedic texts. And in fact, there was even a treatment prescribed for it, Mucuna pruriens, a herb. And it turns out that centuries and centuries later, the active ingredient was identified. It was dopamine. Remarkable, remarkable. So this is the earliest evidence we have of humans identifying this disease. But really, we had to wait till the 1800s when a primary care doctor, James Parkinson, wrote an amazing essay. He was a primary care doctor, not a neurologist. And he just put it together by watching his patients, classical empiric medicine. You can see what he writes here. Involuntary tremulous motion with lessened muscular power, in parts not in action, and even when supported, with a propensity to bend the trunk forwards and to pass from a walking to a running pace, those small, quick steps. The senses and intellects are uninjured. So he wasn't quite right about that last bit, and that's something we'll come back to. Presumably, patients didn't live as long then. Of course, they did not. And some of the more severe features of this disease were probably not noted by him, but he certainly picked up on the motor features. In 1912, the student of Alois Alzheimer, Friedrich Levy, described a finding in the brains of patients with this syndrome. And this really turned out to be the defining pathology of Parkinson's disease versus other causes of Parkinsonism. And we call these now Lewy bodies. And you can see them here. You can see there's a structure here in a neuron. This whole thing is a brain cell, a neuron in the midbrain, and it is accumulating an abnormal morphology here, morphologic feature, which we know as the Lewy body. And in 1961, a remarkable coming together of neurochemistry, principally in rodent models and eventually in humans, really tied dopamine to this disease in an amazing way. And read this description from the 1961 paper. Bedridden patients who are unable to sit up, patients who could not stand up when seated, and patients who when standing could not start walking, performed all these activities with ease after levodopa. This is a precursor for dopamine. They walked around with normal associated movements, and they could even run and jump. The voiceless, aphonic, that means loss of voice, speech, blurred by palilalia and unclear articulation, became forceful and clear as in a normal person. So this was really an amazing story. There's really nothing that works for a neurodegenerative diseases like dopamine. The flip side is, of course, everyone started tying dopamine to Parkinson's disease. It was natural. This drug had a remarkable effect. But as we'll see during this talk, there's so much more to Parkinson's disease than just the loss of dopamine cells. But these were really important findings, and they did tie Parkinsonism to the area of one of the areas of the brain, the midbrain. This is a slice through where you would find the midbrain, and you can see there's a dark substance here called, literally in Latin, the substantia nigra, the black substance. And in Parkinson's disease, you lose that pigment. This is the normal case here. And this is where, so microscopically, this pigment is what you're seeing in these cells that secrete dopamine. So dopamine, the substantia nigra of the midbrain, were squarely tied to Parkinson's disease by this time. And really wonderful work that then followed started to identify how dopamine might affect our movement. I'm not going to go through in any detail the circuitry of what are called the basal ganglia, the deep brain gray matter nuclei. But I will just say that there are some really important circuits here that go from the top of our brain, the cortex, deep into the brain, into these basal ganglia, and come back out. And these cells in the substantia nigra that secrete dopamine provide dopamine to this circuit and regulate it in really important ways that facilitate the rate and rhythm of our movements and even our thoughts. So when you lose dopamine, you don't just lose movement, you lose executive function and other attentional features, other really important aspects of our cognition. So these circuits are extremely important. And what you're seeing here blown up is what a dopamine neuron might look like when it projects into these basal ganglia. So the basal ganglia neuron is here, the dopamine neuron is here, and you can see there are packets of dopamine in what we call vesicles that release their cargo so that the next neuron in the basal ganglia can take it up. We call this area the striatum, and I label it here. And many of you will know that we can monitor the dopamine transporter. You can see that there's a way in which we take up dopamine in the dopamine neuron when it projects into the striatum. We can create a scan through what's called a ligand that binds the dopamine transporter so we can see it light up in the brains of living patients. And what you see here on the left is a normal scan. It's comma-shaped, and you can see it's starting to become abnormal here on the left, which is really the right side of the brain. You can see this comma shape starts to look more like a period or full stop. So that is what we can visualize in life, and that's what you're really seeing is this underlying circuit dysfunction that we call striato for striatum, nigral for the substantia nigra dysfunction. And when that circuit is impeded, we have a loss of these critical motor and cognitive functions, that constellation being known, as I mentioned, Parkinsonism. So then in our historical tour, 1983 is the next place I want to stop. Bill Langston wrote a remarkable paper in Science with colleagues showing that Parkinsonism could actually develop very quickly in patients who were using certain intravenous drugs. And so this was a remarkable paper in which he describes 4 people that developed marked Parkinsonism after using an illicit drug, and they ended up finding what the active ingredient here was, the toxic ingredient. It was known as MPTP. He and his colleagues described striking Parkinsonian features And in the pathology, they also noted that it selectively damages cells of the substantia nigra. So this was the first proof that if you just took these cells out, you get Parkinsonism. Not Parkinson's disease, but Parkinsonism. It was a very important finding. And so you can see here, years later, they did DAT scans on these patients and they described the findings here. And you can see the depletion. This is normal and this is depleted. And so this was a very important finding. It enabled the first toxic models of Parkinsonism to be made in the lab, in animals. And it solidified a belief in the time, so between the 1980s and 1990s, that Parkinson's disease must also be an environmental disease. And so it was very firmly thought of as an environmental disease. And this story of dopamine, I'm going to call it, epoch zero here, the first 2,500 years, had another remarkable moment in the 80s and 90s when Benabid and colleagues started stimulating the brain and showing that they could dramatically change Parkinsonism through stimulation with electrodes. And this eventually became the therapy deep brain stimulation, which is a very useful therapy till this day, as is, of course, dopamine. And this is a nice image from a New England Journal case history showing how this in principle works. Essentially, an electrode is placed into the brain that stimulates, and the net effect of that is inhibition, although the effects are quite complex, that targets the basal ganglia circuits that I mentioned before. So one of them was called the globus pallidus, and there was another related structure called the subthalamic nucleus. Essentially, what this treatment does is it attempts to bypass the need for dopamine. So patients need less dopamine because the circuit is being manipulated. That deep brain circuit is being manipulated directly through an electrical signal that's sent from the pulse generator. Now, the era of dopamine, of course, continues. We are innovating in this area all the time, and there are interesting new technologies. For example, surgery-less focused ultrasound can now be used to target some of the deep structures like the subthalamic nucleus and treat Parkinson's. We can talk about this more in Q&A. None of these treatments is perfect. They all have their pros and cons, but they are remarkable. They make a significant difference to our patients' quality of life. This is a disease that can be treated very effectively for a decade or more in most patients. And I'm going to close this era of Parkinsonism with a 2001 paper showing that you could take embryonic dopamine neurons and transplant them into the brain to replace those dopaminergic cells. And here you can see evidence. This is the needle track. This is just showing where the needle track was of this transplanted material from embryos. And you can see that it's darkly pigmented. It's looking like that black substance. These are bona fide dopamine neurons that are forming connections all through the brain. So this was also, a landmark study. In 2022, we're now seeing the next generation of these trials, multiple in Europe and in the US, starting using more sophisticated techniques of making stem cells from patients. So this was really the story of dopamine, and where this first phase takes us is really to some remarkable symptomatic treatments for this disease. So now I'm going to move through fairly quickly, so apologize for that. Time is limited. And we're going to talk about the next 20 years or so. Because the next 20 years have been all about trying to slow this disease down, prevent it, reverse it, not just give symptomatic treatment to patients, but change their outcome. That's the key, right? That's what all our patients want. And I'm going to divide this into 3 epochs. Epoch 1, the era of alpha-synuclein genetics and what I'm calling cell autonomous mechanism. The cell autonomous just means what is the intrinsic property of dopamine neurons or other neurons that makes them vulnerable, and how do we fix that vulnerability? The second aspect is multi-system disease, this idea that, Parkinson's is not just a disease of dopamine neurons. There's a lot going on here. How does this pathology occur in so many different cell types? How does it move from one cell to the other? How do we stop that spread? That's epoch 2. And epoch 3 is what's happening outside the brain. Can we unlock different types of treatments? So we'll talk about each of these. We'll spend most of our time on epoch 1 because we're actually still in epoch 1. Epoch 2 started a lot later, and epoch 3 is just starting now. So epoch 1, synuclein. So it turns out that there was an intense focus on what this Lewy body was. For 100 years or so, almost 100 years, we knew this was a feature, but we didn't know, was this a bystander event? Was this really an important event? What were these Lewy? What was the implication of having Lewy bodies? Were they important? So what's that culprit protein? Well, it turns out this is a remarkable story of the debt that biochemistry owes to molecular genetics. This is a really amazing story because in the late 1990s and early 2000s, these amazing patients who were volunteering for research, like the woman I showed you right at the beginning, we were starting to, the field, this was a few years before I got into neurodegeneration, but this is one of the major driving forces that got me in. Mutations were starting to be identified in these patients. And so this wonderful neurologist, Larry Golby, a friend and colleague, started identifying patients first in New Jersey and turning the Parkinson's world upside down, because remember, at that time, everyone thought it was an environmental disease. He started shrewdly observing that this, in some families, was passing generation to generation. There must be a genetic component. And so mutations in the alpha-synuclein gene were identified. These were the first known mutations for Parkinson's disease. And on the basis of this finding, this protein in Lewy bodies was identified as alpha-synuclein, all in the same year. Really remarkable. And so what is synuclein? We're not going to go into, as I say, much molecular detail, and we're discovering new functions all the time. But alpha-synuclein is a remarkably interesting protein. It is a couple of % of brain protein. It's a very common protein in the brain, which means that it's always at risk of aggregating, forming the wrong kind of shape. So it often is unfolded when it binds to membranes. Remember, I told you vesicles that were holding neurotransmitters. Synuclein binds to the outside surface of those vesicles. It has a remarkable conformational change. It changes shape when it does that. And sometimes this high abundance protein forms the wrong kinds of shapes. And these have different names. Oligomers, here I'm showing them as amyloid fibrils. So that's the word I'm using, fibrils. But essentially, this protein is associated with vesicles, and one of its functions is in the transport of those vesicles. Now, it turns out that the woman we talked about at the beginning had a remarkable family history too. Every generation of her family, roughly 50% of the people had Parkinson's disease or a related dementia. And it turns out that her family taught us so much because they didn't have a mutation in the gene. They had extra copies of the normal synuclein gene. So just extra copies of that gene that would lead to increased levels of the protein was sufficient to cause disease. And this meant that models of this disease, now the first genetic models, not environmental models, but genetic models could be made. And I'm not going to go through this history. It's a wonderful history. But people started expressing synuclein in neurons in the lab, in worms, in flies, in mice, and even in living test tubes yeast cells. I just highlight the contributions of my PhD advisor, Mel Feeney, and my postdoctoral advisor, Sue Lindquist, for creating some of these first pioneering models of Parkinson's. Mel's paper in 2000 is really remarkable. She shows that if you express synuclein in a fruit fly, it stops climbing and it develops synuclein aggregates in its dopamine neurons. It's a really, really cool paper. If you haven't read it, I recommend it. And Susan's work suggested that even cells that evolved billion years before us could be really informative in highlighting the biology of what happens when synuclein clumps and forms those abnormal shapes. When I was a junior resident at Mass General and the Brigham, there was another breakthrough. Shinya Yamanaka showed us that we had an avenue to creating a human model. This was a landmark paper, and he won the Nobel Prize for this in 2012. You all know that embryonic stem cells can form any cell in our body: cardiac cells, nervous system cells, immune cells, you name it. And what Yamanaka and Takahashi showed us was that you could take an adult cell like a skin cell, and in a way that was much easier than was expected, you can turn that cell back in time into an embryonic stem cell-like cell that they called an iPS cell, an induced pluripotent stem cell. So this was remarkable because at this time, the only way we had an avenue into the brain of our patients was after they died, or with something like a DAT scan, but nothing specific. For the first time, we could theoretically make brain tissue from a living patient, just like our cancer colleagues have been doing from biopsies. We could do that, and this was a remarkable moment. And so we did that, other people did that. We were among the first to start making cells relevant to Parkinson's disease from these iPS cells. And I teamed up with Sue Lindquist, Rudolf Janisch, and others, my close collaborators, Chi Chung and Dan Tardif. And what we did was we took cells from a patient, the same family that Larry Golby had described. We took her cells, which had the alpha-synuclein mutation, and we used genome editing. You've probably heard of CRISPR, but this was the grandfather of CRISPR, called zinc fingers. There was no CRISPR at this time. This was in 2007 or 8. And it took 2 years for Frank Soldner and Rudolf Janisch's lab to genetically correct this mutation. But he then gave us a very, very powerful reagent, because he gave us a stem cell that had the mutation and a stem cell from the same patient that had the mutation corrected. That means we could start to discover the biology of the human synuclein gene in a human cell, and we could start to figure out how to reverse it. And so we actually, at that time, no one thought we'd be able to find clues. We actually genetically corrected the cells, and we used that yeast test tube to express synuclein, and we found drugs that could do the same thing as correcting the mutation. So here's a yeast cell that's forming aggregates. Here's our patient from whom we made these iPS cells. We now have her brain cells, and we could start to discover drugs. And that's what we did. And I'm going to start moving fairly quickly now, but we found a variety of drug-like molecules, including one that's now in clinical trial. This was in collaboration with my colleagues at Humanity Therapeutics. And we have found a new class of drugs that can change the property of vesicles. That means that alpha-synuclein is less sticky to them, and this seems to have an ameliorating effect. And we went from a hit in a yeast screen to clinical trial in less than 5 years, which is remarkable. And this is actually mini brains in the lab from this same patient that I've been talking to you. And you can see, as we make these mini brains, abnormal synuclein, the green gets brighter where there's a mutation. It doesn't get brighter when we genetically correct it. And the graphs above just show that we can genetically correct these, we can mimic this correction with the drug that I just mentioned. And that's now in phase 1b clinical trial. So over the last 2 decades, there have been other genes that have been found. You might have heard of the Gaucher-related gene, GBA, LRRK2. There are many others. And mutations in these genes or in their vicinity, for those who are more technical, common variants, rare variants, have been identified. How heritable is Parkinson's? It was thought of environmental. and now we think of it as we're very genetics focused. What's the reality? Well, when we look in twins, there's about a 20% concordance rate in twins that have exactly the same genetic code. But there's a 13% concordance rate in twins that are dizygotic, that do not have the same genetic code, suggesting the environment's also very important. There have been some recent wonderful papers that have told us to beware of the latency of Parkinson's disease. Just because it starts in one twin and doesn't start in the other does not mean that other identical twin is not going to get it later in life. So when this group looked very carefully at 5 sets of twins that were thought to be discordant, not agreeing for their Parkinson's disease risk, when they took a deep dive on the patients that didn't have the disease, they found evidence that they actually did. It was just taking longer to develop. So we think the heritability is at least 30%, probably more, but we don't know. And most of this heritability, even after 20 years of research, most of these inherited factors are not known. And there are large genetic studies underway now to address this. So at the end of Epoch 1, where are we left? Well, we're left with some really important insights into the cause of this disease, what makes cells vulnerable to having this disease, and a set of drugs. I showed you a class of drugs called the SCD inhibitors because I was working on them, but there's many other classes of drugs that have been developed targeting alpha-synuclein, including now genetic therapies like antisense oligonucleotides that lower the level of alpha-synuclein. For LRRK2, another gene, there are now inhibitors that affect the function of LRRK2, its kinase activity. For this other enzyme, glucocerebrosidase, the Gaucher-related protein, there are now drugs that modulate the enzyme. But most patients don't have a clear genetic cause. Almost everyone who walks through my clinic door is not in one of these categories. So even though it is very exciting, we have these trials, we need to do better. So Epoch 2 now, we're going to move from what's happening within a cell to the fact that this is really a multi-system disease. And I'm going to just make some observations here. This is the clock that this woman drew in January 2000. This is 2002, and this is 2003. Look at the change. She was asked exactly the same instruction. This is not just a motor disease. This is also a disease of cognition. Now, granted, this patient comes from a family with a very unusually strong form of Parkinson's disease. But one of the key debilitating features late in the course of this disease is cognitive dysfunction and dementia. So we shouldn't just be thinking of this disease as a motor disease. And I'm not going to read through all this text, but when you look at her clinical history, this is this woman's clinical history, there was not just physical slowness, but mental slowness. She had tremor. She had what's called RBD. She was acting out her dreams. She had loss of weight. When she stood up, her blood pressure dropped. Her mini mental state exam was a 13 out of 30. She had lost her sense of smell. So there was so much, and towards the end, she was incontinent. There was so much going on with bowel and bladder, sleep, other features, cognition that are not related, directly to dopamine. And so in this epoch, we think of Parkinson's disease as a multi-system disorder, and Brack and colleagues in 2003 started describing a staging system of Parkinson's where they said this disease may spread from one part of the brain to the other. It may actually start way before dopamine neurons. It may start lower in what we call the brainstem and then affect dopamine neurons, ultimately affecting many parts of the brain. So this was, again, superb clinical medicine and pathology observational skills from Brack, who described this. And this is the type of pathology you see all through the nervous system. These are dopamine neurons, these are cortical neurons. So this is widespread pathology that affects many cells. And then there was another really interesting finding that came several years later. Remember I told you that patients had been grafted with embryonic stem cells? They grew into dopamine neurons. I should say they were grafted with embryonic dopamine neurons. Some of these patients did well. Some of these patients had terrible side effects. So this was never FDA approved. Years later, when their grafts were examined, it was noted that they also developed alpha-synuclein pathology. Another very important observation suggesting that maybe this pathology can spread from an abnormal part of the brain to a part of the brain that was normal and is now accepting that pathology. So somehow these grafts that should have been normal were also, in just a few years, developing alpha-synuclein pathology. And so the field started thinking of mad cow disease and prions and started wondering, could this be related biologically to a prion? Now, I want to be very clear. There's no evidence that alpha-synucleinopathies are infectious diseases like prions. But there is some biology within cells that may be reminiscent. So this is what happens when the normal prion protein misfolds into an amyloid. You get a terrible neurodegenerative disorder. This is what happens in humans. It's called CJD. This is what happens in sheep. It's called scrapie. And Stan Prusner had discovered prions back in the 80s, where he described a remarkable property that a normal protein could meet an abnormal prion protein, and that abnormal protein could make the normal one abnormal. This was just a protein that was acting like an infectious agent, so we called it a proteinaceous infectious particle, in other words, prion. And so the idea started to develop that maybe in Parkinson's disease, the spread that Braque had described could be related to spread from cell to cell. And that could be that alpha-synuclein was being released by one cell, taken up by another, and then forming abnormal pathology in that recipient cell. And this is described here, that alpha-synuclein can spread along neural circuitries. This is a review that we wrote on this, and this is really for those who want more detailed explanations to read. But now the cell biology of spread is really evolved even since the time we wrote this review. And there are many mechanisms through which alpha-synuclein may pass from cell to cell, and this is a very active area of investigation. One of the other things I mentioned was bowel dysfunction, bladder dysfunction, sweating dysfunction, dysfunction of blood pressure. These are autonomic functions of the nervous system. And it turns out all of these, or many of these functions, are affected in synuclein diseases, including Parkinson's disease. And there is alpha-synuclein pathology that has been found throughout the nervous system. In this remarkable paper from Ted Dawson and colleagues that just was in Neuron a few years ago, they showed that if they just inject synuclein fibers into the gut, so think about constipation and those features, if they just inject abnormal synuclein into the gut of mice, that synuclein can spread up the nerve that we call the vagus nerve into the brainstem and create pathology in the brains of those mice. So there was spread from the gut to the brain. And if you cut that nerve, you don't get the pathology, suggesting that there was either some kind of direct spread or there was a response of recipient cells to the toxic protein that ultimately led alpha-synuclein to aggregate in the brains of those mice. Now, if they remove synuclein from the brain, they use a mouse that doesn't have synuclein, they get rid of the pathology. So this was a really elegant study that started establishing some of the key principles that had been observed in the pathology. And over time, it's become clear that alpha-synuclein folds into different shapes. This is very important. It doesn't just, when it misfolds into what I called an amyloid fibril at the beginning, it can form different shapes. So when you look at it in the electron microscope, or with even more sophisticated forms of microscopy, you can see it forms different shapes. And it turns out that alpha-synuclein can form many different types of shapes, and its effects in different cells as it spreads from cell to cell are different. So in other words, alpha-synuclein can make different shapes in different cells of the brain, and in turn, those brain cells can respond differently to those different shapes. This is a really important finding, and we call this the host strain problem, to borrow a term from prion disease. That the host cell, what I was calling before cell autonomous, the vulnerability of those cells is really important, but the toxic form of the protein is also important. And as many of you will know, there is a family of diseases that cause Parkinsonism that are all related to the misfolding of alpha-synuclein. And it turns out that different shapes may be partially responsible for these different diseases, because if you just make these different shapes in the lab and inject them into a mouse brain, you get different pathologies. So something really important is encoded in the shapes into which these proteins misfold. And you can measure these in the lab. Claudio Soto's lab 20 years ago in the prion field published a remarkable experiment that I show here on the top right, that you can take proteins and you can take a normal protein and an abnormal one, and you can grow these amyloid fibers in the lab and amplify them. So now we can take synuclein from the skin or the spinal fluid from our patients, and we can amplify it. And what we're seeing is that patients who have Parkinson's disease, you can see those in red, their synuclein amplifies in a different way to patients, for example, who have, in this case, in blue, multiple system atrophy. So this is really important. Okay, so here's an example of a family we follow where there's Parkinson's and MSA in the same family. You know, we take a deep dive on these patients in our movement disorders genetics clinic, and we try and understand what the genetic causes are of these incredibly interesting families that show different pathologies. This is one of those patients, and in this case, you can see alpha-synuclein in the skin neurons, in the autonomic cells in the skin, and the patient with MSA progressed faster. She died. We confirmed this pathology. So in the movement genetics clinic at the Brigham, we study families like this to learn more about what's happening. And this spread idea has really been transformative. I don't think it's the whole story, but I think it's transformative in understanding how spread might occur from cell to cell, how that might explain the circuit dysfunctions that happen in Parkinson's. And at the end of this epoch, we really talk about spread being blocked by an alpha-synuclein specific antibody. Is that possible? Could we use an antibody that blocks alpha-synuclein as it spreads and slow the progression of the disease? So many of these are now in clinical trial. So what we end up here with at the end of this epoch is really a new set of therapies, again, aimed at disease modification. Now, very briefly, what else is going on outside of brain cells? The brain is an amalgamation of many different cells. We tend to think about neurons a lot, the electrical cells, but there are glial cells that have extremely important function, especially in neuroinflammation. And I won't talk about this in detail, but microglia are getting a lot of attention these days as modulators of brain inflammation. And maybe we could modulate inflammation to modulate the outcome. In this remarkable study that was published in Cell a few years ago, that I'm not going to go through in detail, but what was shown here was if you just had the normal microbes in the gut of a mouse that was expressing synuclein and had a synuclein disease, well, you got a Parkinson's-like syndrome in those mice. But if you gave those mice antibiotics, got rid of all the bugs in their gut, you could get rid of the disease in their brain. So just changing the microbes in the gut could change the outcome in the brain. And if you put Parkinson's disease microbes into these mice, they did worse than if you took microbes from patients without the disease. So a remarkable and landmark study suggesting that we might be able to alter the microbiome to change what's happening in the brain. And the mechanism here, and you can see it in the middle, is that certain factors released by these microbes can change the activity of those neuroinflammatory cells in the brain. So this is just for your reference, but I'm not going to go through this right now. It turns out these microglia can even take up synuclein and pass synuclein from among themselves. And this was just a recent paper suggesting that there's a lot more going on in the brain among this cellular milieu than just the effect of aggregation in neurons. So at the end of epoch 3, which we're of course in the midst of now, we are thinking of modulators of neuroinflammation, including changing the microbiome and metabolites that could give us external ways, easier ways to treat the disease than simply manipulating the brain. And finally, in the last few minutes, I'm not going to have time to go through all of this, but finally, I want to kind of tell you where we're at right now and what period I think we're in right now. I think we're in the period of understanding that a one-size-fits-all strategy may not be sufficient for treating Parkinson's disease, that this disease is complex, it's heterogeneous, and we need to, like our colleagues in cancer, we need to use all of the technologies, genomics, stem cells, and so forth, to understand the subtypes, the subgroups of Parkinson's disease. This is, I think, a really important area of research. And I'm going to really just put it another way, that we may not be able to prevent or modify the outcome of patients with these diseases because there could be differences among our patients, a one-size-fits-all strategy. Either our biological insights may be flawed, or the clinical trial designs may be flawed. If we're mixing these people up, we may be treating some people in our trials, but we would never know. Or of course, we have to learn on both sides. Samaya Nazin in my lab is really looking at the Michael J. Fox Foundation cohort right now in great detail. This is a remarkable resource in which stem cells and genomes have been collected for a group of 500 or so patients that have been followed over time. And when you take and look at these patients and look at the different ways they progress, you can see that some patients within just a couple of years of diagnosis progress very fast, and other patients don't progress fast at all. So one way in which our biology may be flawed is we're targeting vulnerability factors that modulate risk to this disease, but are they the same as progression factors? Maybe we should be targeting progression factors rather than vulnerability factors. This is an open question. Our clinical trials may be flawed. We may find ourselves in a situation where these patients depicted in orange and these patients depicted in blue are mixed up in a clinical trial, and now we're expected to see in a year if they respond to a drug. But these patients, you might have a chance to see a response. These patients, you'll never have a chance. So if you mix apples and oranges here, your clinical trial may be flawed, even though you have a perfectly good drug. So this is a challenge. This is exactly another way of depicting this. We're mixing apples and oranges here. We give some people a treatment, some people a placebo, and not surprisingly, if their biology is truly heterogeneous, we don't see effects in clinical trials because we're mixing these patients up. They may have different underlying biologies. So the alternative that we're working on, and others in different centers, is we are tracking our patients very carefully in time before we give them a drug to try and understand whether they're responders or non-responders. We use their own history as a control for their future. The effect of the drug is compared to their prior trajectory. And so we're trying to break down the biology here, because if we have responders to a drug or non-responders, we're going to need to understand why is that the case? Why did some patients respond to this drug and some patients not? We need a mechanism. And so we're integrating genetics, we're integrating this idea that synuclein folds into different shapes, and we're taking into account environmental exposures to create the next generation of stem cell models from our patients that have been carefully tracked in the clinic from whom we have biospecimens and biomarkers and so forth, and for whom we can then create a mimicking model in the dish, okay? So this is very active here. We have 50 clinicians and scientists in our division working on this, doing exactly what I just described. Two major labs, the Scherzer lab and the Khurana lab, are working on this. The Scherzer lab is leading the Harvard biomarker study, tracking 3,000 patients over time, is recently starting to uncover genes underlying Parkinson's progression, a very important set of studies. In our group, we are taking novel genomic and artificial intelligence methods. We're using the biology of synuclein to understand what those molecular subtypes in our patients might look like. We're making brains in the dish and even cohorts in the dish where we're mixing cell lines from multiple patients and pooling them together so that we can finally achieve clinical trials in a dish paradigm for these complex diseases. And we're building other cohorts, for example, in multiple system atrophy. This is a picture of the group. I wish I could talk about all of their work, but it's a great pleasure, and it takes a lot of dedication and hard work to do human stem cell work. One of the, and I'm going to take you through in just the last couple of minutes, I'm going to take you through some of these approaches. So one of the targets here is genetics. Most of our patients, I told you, do not have a known genetic cause. How do we think about their complex genetics? Well, when synuclein aggregates, it aggregates in a cell that is packed full of proteins that are surrounding that protein as it aggregates. My lab is developing methods to find out what those proteins are, using sophisticated screening technologies to understand when this protein mucks up, what are all of the proteins in the cell that are really important to that misfolding protein biology. And we've created the first roadmaps of synuclein, connecting it through these different methods to other proteins and genes in the cell. A lot of these genes that we find, a handful of them anyway, are known Parkinson's genes, found here much more likely than you'd expect by chance. So what we think is that other patients also live in these maps, that they have genetic factors related to the misfolding of these protein, and if only we could find them. And so we're developing genetic and AI tools to do that. We're doing that in conjunction with Shamil Sanayev's group and Jen Peng's group at the University of Illinois. Shamil is here local. He's a professor of genetics at this medical school. We're also trying to make better models. I talked to you about yeast cells. I talked to you about mouse models and simple human stem cell models that we made a decade ago. But now we can do better. We can rapidly make neurons of the right type at scale in the lab, and we can introduce into those cells the right types of aggregates from those patients. I told you something really important is encoded in the shape of those proteins. We can now capture that and put that into these brain cells to create much better models of what's happening in the brain. So you can see our neuronal models on the top, a matching brain on the bottom. We're getting much better at making a more faithful replica. We can watch these aggregates form in the dish, and we've used this recently. This is unpublished work to find new types of aggregates in the human brain. We're looking at environmental toxicants. Beata Ritz and Kim Paul are collaborating with Edinson in my group, Lee Rubin, a professor at Harvard, and Rich Królewski, who works between both our labs. These guys have done a remarkable job from UCLA, where they have quantitated environmental exposures and shown for the first time that indeed pesticides and other toxicants are important in Parkinson's risk. So we're seeing these two fields of genetics and environmental biology converge now. But we need models to understand what's the environmental contribution, what's the genetic contribution. And human stem cells in our lab are providing a mechanism to do that. So we're making brain cells from patients in these studies, and we're trying to understand the relative contributions of the toxins in the environment and their genetics. And as I mentioned, we can make now whole groups of cell lines. So we could imagine taking 100 or 200 patients' lines and growing them in the same dish. So this is really remarkable, and we're pursuing this technology in collaboration with Lorenz Studer, a stem cell pioneer at Sloan Kettering. So the vision here is matching what we're doing at the Brigham in the clinic. We would love to be able to create these replicated clinical trial cohorts in the dish where patients who have no known gene mutation are aggregated into different molecular subtypes, and innovative stem cell technologies allow us to create a cohort in the dish so that hypotheses can directly be tested in brain cells we make from our patients, eventually at population scale. And finally, I'm going to return back to the families that we care deeply about. I started with a woman who was fired from a job because she had an alpha-synuclein mutation. Now we follow a whole family in northern Spain that have alpha-synuclein mutations, and they have a remarkable story. Every generation, some of them escape the Parkinson's, some of them escape the dementia. They all get the peripheral autonomic problems, for example, changes in their bowel and bladder, but they do not get the motor or the cognitive manifestations. The question is, why not? Why does this patient in purple progress so quickly, but his first cousin, who's the same age, hardly progress at all? We have cases in this family where a 30-year-old son has got Parkinsonism and dementia, but his father with the same mutation is perfectly well. So we are now taking these patients, this is just a DAT scan to illustrate it, we're taking these patients, sequencing their genomes, making stem cells from all of them, taking their blood, their spinal fluid, their microbiome, putting that microbiome into matched transgenic mice that we have in the lab. We're trying to understand, trying to take these families who've donated their time to understand if they can unlock some of the mysteries of why Parkinson's progresses so quickly in some and is so slow in others. So here are my last remarks from within Epoch 4, that the differences among our patients may be a bottleneck for therapies. A systematic cellular survey of the consequences of protein misfolding may actually guide us to stratifying our patients into groups. That patient-specific stem cells offer a surrogate pathway to tissue that we can create now in the dish. And that we shouldn't think just within neurons, that outside of neurons in glial cells and outside of the brain in other organs, there may be critical clues for slowing down this disease. So I thank you very much. And I know I went a few minutes over, but I'd be very happy to take questions. And thank you again to the Harvard Brain Science Initiative. Yes.
In the example where you said the son had the disease to a great degree that the father didn't, if you were to just sequence those two individuals and compare gene by gene, a lot of genes, 20,000 genes, wouldn't at some point be had enough of that type of data to be able to identify protective genes or resilience genes?
So that's exactly right. And so that's why we've sequenced all of their genomes. But you also were right about saying 20,000 genes and just a handful of patients. We are not powered to find those genes because there are too many possibilities. So we need biology to guide us. And so that's why instead of looking at 20,000 genes, our group is saying, can we narrow the search space to just a few hundred genes that we think are really important for this pathology? And that's why we do all of that experimental work in the lab to narrow down a set of genes that we think, are where the money might be. The other thing, Ofer, is you can think about that very interesting study in the microbiome showing that you can change the outcome in the brain by just changing the gut microbes. So it could be an environmental effect. There could be something else going on. So for this family, we're trying to do all of the above. We're trying to take it from different angles, but we absolutely have sequenced their genomes. There was a question over there first.
The very question that I had was, assuming that alpha-synuclein is expressed in all cells of the body, so what is it about the cells that is especially?
that's a great question, but I think we should turn it around a little bit. Firstly, synuclein is not expressed everywhere. I just want to be clear on that. I didn't elaborate. It's expressed most highly in brain cells and in neural cells across the body. So when it aggregates in the gut and in the skin, it's actually within nerve cells. Having said that, it's also found at high levels in blood cells and kidney cells and some other cells, but it is really abundant in the brain. And I think then you're asking a question of within those neural cells, what explains the differential vulnerability of some groups of cells versus others? And that's a really important question. I think it's just important to know that a lot of different neural cells are affected, and we shouldn't be too dopamine-centric. But there have been some great studies on this from Ulla Isaacson's group, Jim Surmeyer's group. I can give you those references. One of the approaches they've taken is taken the dopamine neurons that die in the disease and the dopamine neurons that don't die and asked, what are the differences? And so it turns out that, that there's a nice convergence here. That if you look at dopamine neurons, I'll just give you one example. Dopamine neurons that don't die, they have a much better way to buffer the metal calcium. And that causes them to resist calcium-related fluxes and stresses. So they have a lot better mechanisms for protection, and calcium is just one, but a variety have been identified. In fact, even in a yeast cell, this is where there's convergence. When you introduce alpha-synuclein, one of the first changes that you see is an abnormal change in calcium regulation. And so if you're a brain cell that has a better way to deal with that, you might resist the pathology. That's just one of a number of different mechanisms that have been described to look at differential vulnerability. But these studies are also, they're old in some ways, 20 years old, but they're also still very active studies because, as you say, there could be some really important clues there. I just think it's important to know, and I want to emphasize, dopamine neurons are unlikely to be the first neuron that succumb in Parkinson's disease. This disease starts decades before the patient comes to the clinic. And so there's a lot going on before the patient presents with the motor symptoms. Yes.
Hi, Dr. Khurana. Oh, wonderful. Hello, Dr. Khurana, thank you. You spoke about it probably halfway through about having a hit in yeast that within 5 years you were able to bring it to a clinical trial. Could you just expand a little bit upon that and what were the factors that allowed that quick timeline from bench to trial, and are there things we can learn from that?
Yeah, I think the underlying discovery process there is called a phenotypic screen. You have an abnormal cell and you fix it. The problem is when you fix it with a drug, you often don't know how that drug works. So I didn't go into detail, but in those 2 papers we published in Science in 2013, we provided an avenue to finding the target. And the avenue was if you could make sure that a brain cell was fixed in the same way that a yeast cell was fixed, you could actually use the yeast genetic toolbox to find the target of the drug. So one of the major ways in which we sped up the process was using very powerful genetic tools to identify that. Now, those genetic tools are becoming available in human cells, so we're not going to need yeast for that much longer. We're going to be able to develop these kinds of tools in more sophisticated cells, even in neurons. And so my lab, number of labs are developing those kinds of tools. I think the idea of always having human biology at the center and using human stem cells as a way to quickly show that you're actually perturbing human biology. In this case, we obviously did work in mice and other models too. That was also a way to quickly find something that was potentially valuable to patients.
Thank you for the great talk. I had 2 questions. The first one being, what determines the misfolded state of the alpha-synuclein in the cell? So you mentioned there's heterogeneity in that. So
sorry, what was the last bit? There's a what in that?
There's a heterogeneity in
Yes.
the type of misfolded aggregates they form.
Yes.
What is the underlying cause for these difference? How does it change? And 2 is for antibody therapy, are you tagging it with something that brings in an E3 ligase or something that tags it for ubiquitin proteasome mediated degradation or autophagy, and how are these pathways affected? Because these are usually the pathways responsible for clearing these aggregates, right?
Right, great question. So first one on the polymorphic strains, how do they form? What's the mechanism? Short answer is the environment in which they form is important. We know that. We know that, for example, this is Virginia Lee's work, we know that multiple system atrophy strains, for example, form in glial cells, in oligodendrocytes. So there is something in the oligodendrocyte that is important in creating a conformer that is MSA specific. It doesn't, if you passage that strain through neurons, you don't generate that conformation. So we know there's environmental factors within the cell that are really important. That's what I referred to as the host-strain interaction. We know that the backbone of the protein is pretty important, although in most cases, it's the wild type protein. It doesn't have a mutation, but certainly mutations in the protein. For example, in that family I just showed in Spain, they have a very interesting mutation that changes the conformation. So there is intrinsic properties within the protein that could change it, and there are environmental factors as well. What those factors are, we're really just at the cusp of discovering.
On that, so do you see the same misfolding in the same type of cells, or is it also varying?
That's really important question. So the reason we're making these new models from human stem cells is to understand this bewildering diversity of inclusions that we see. They're not all amyloids. I mean, this was a simple talk. Some of them are membrane rich and they don't have amyloids at all. And one of the major projects in our group is to understand what the consequences are of all of these different types of inclusions that form, because there's a group of them. And we tend to think of them very simplistically as all one thing. They are not one thing. They have different biology. Some of them could even be protective and some of them detrimental. And that has implications for how you make a model in the lab, because you don't want to be targeting something that's protective. So this is a very active area of research for us. And then your other question was, are these antibodies intracellularly active or extracellularly? At the moment, the clinical trials are to target the extracellular synuclein. So there's an open question. So they're not using any kind of downstream mechanism for degradation. They're not targeting intracellular synuclein, but there are a variety of different methods that get into cells that are actually being used to create, exactly as you said, to facilitate the degradation of synuclein. And those are kind of engineered approaches, intracellular approaches. So I didn't talk about those today.
Hi, I've heard a lot of people talk about how mitochondria are somehow important in Parkinson's disease. Can you talk a little bit about how that is?
Yeah, it's great. So there are certain big aspects of Parkinson's I didn't touch today. Some in some ways because they're very well known, but they were behind some of the data I showed. So that paper in the 80s by Bill Langston that showed the intravenous drug uses that MPTP was a toxin, that really opened up the field to this idea that energy metabolism in dopamine neurons is really important. And that if you mess with it, so for example, an environmental toxicant that's not an intravenous drug, but something like paraquat is a pesticide, if you mess with mitochondrial enzymes, you really have a situation where dopamine neurons are vulnerable. Now, I focused a lot today on molecular genetics and kind of what's happening maybe at, let's call it upstream of that. But if you then start digging deep into how these toxicants are working or the downstream mechanisms of alpha-synuclein toxicity, the mitochondria are a major target. So all of those steps in between are not fully elucidated, but there are a variety of different cascades in the cell that converge on mitochondria, and I'd be happy to talk in more technical detail about these, that reflect probably a really important aspect of the biology. There are also gene mutations, and I could have mentioned them. PINK1, Parkin, in case people study Parkinson's disease, they're very interesting. They often don't develop synuclein pathology. They don't often develop classic Parkinson's disease, but they develop Parkinsonism and they lose their dopamine neurons. And so there's this constant dichotomy between what is toxic to a dopamine neurons, where mitochondria are clearly very important, and what is central to Parkinson's disease itself. That's not just a loss of dopamine neurons, it's a loss of multiple neuron types associated with alpha-synuclein aggregation. So that's a convoluted answer in some ways. I hope it's at least generally helpful, but there are genetic causes of Parkinsonism that pinpoint mitochondria. There are environmental toxicants that pinpoint mitochondria, and there's a wealth of cell biology that converges on that organelle as being critically vulnerable, especially in dopamine neurons.
I can ask some questions that are on Zoom.
Oh, sure, yes.
So you might have touched on this one a little already, but someone asked, could you speak to advances in treatment, what's coming down the line for Parkinson's that's caused by genetic mutations such as GBA?
Yeah, I think that there are a variety of different approaches for GBA that are being thought about right now. These include genetic approaches, and they include non-genetic small molecule, drug-like molecule approaches. There are some open questions about the relationship of what we should exactly be targeting in GBA-related Parkinson's. Early studies by Dimitri Krantz and us showed that alpha-synuclein and GBA were very relatable. So, for example, in the stem cells that we made from patients with synuclein mutations, one of the proteins that was not being appropriately transported in the cell turned out to be the GBA protein. And we at the time depended on Dimitri Krantz's insights in stem cells from patients carrying the GBA mutations to make that connection. So there is some deep connections between GBA and synuclein. The details are not fully fleshed out. And there are some methods that increase the transport of that protein to where it's meant to go. Ultimately, it goes to the lysosome. So to facilitate its transport to the lysosome, there are molecules that can do that. There are molecules designed to increase the enzymatic activity of GBA. It's still, there are questions about whether that's exactly the right thing to do, whether it's its transport or trafficking, we call it, or its activity that is really the best target. But there are really interesting modulators of this that are now approaching clinical trial. And so we're starting to see a number of companies with products in that area.
Thank you so much. So we have a question on imaging. How can MRI-based techniques, T1, DWI, or fMRI, help in the epoch 4 of understanding and treating Parkinson's?
That's a great question. There are now more sophisticated MR techniques. One I think that's really becoming prominent is automated volumetric. So don't just look at the MRI as two-dimensional, look at it as three-dimensional, automated volumetric MRI, which gives you, I actually showed a couple of images of that, but I brushed through them, but it gives you the volumes of different parts of different brain structures. This is not the most sophisticated technique, but it's starting to enter at the point or get to the point where it could be useful for clinical trials, for subdividing patients into different subgroups and for tracking their response more finely than what we can do in the clinic. The questioner also asked about DWI, ADC. These are different MRI approaches, and we're starting to see some papers that use diffusivity in MRI and other more sophisticated assays to distinguish, again, subgroups of patients that are more likely to progress, less likely to progress. Functional MRI is really important for understanding the underlying circuit biology. I haven't seen that yet really coming to the fore in the context of routine tracking or clinical trials, but no doubt there'll be advances there as well.
Thank you. We have two more Zoom questions. Do different strains of synuclein spread at different rates or have different preferences for where they end up? If so, have intrinsic properties of these strains been attributed to these features?
Another great question. Yes, if you, one of the first papers on this by Pellets and colleagues in Nature showed that if you just create synuclein, purely recombinant synuclein, so not biologically derived, but you make it in the lab as a pure protein, and you just incubate it in different buffers, you can create different shapes of synuclein. And if you inject those into mice, you get different diseases that are stable. So that suggested that the conformation of the protein is important. Now we're at the point where we're taking synuclein extracts from patient brain, or as I mentioned, spinal fluid and skin, and we're able to amplify it in the lab and do the same experiment. And those experiments also suggest that the properties of the synuclein are very different. For example, the MSA, which is a much more aggressive disease, the MSA strain of synuclein appears to be much more transmissible and seeds much more avidly than the Parkinson's disease strain. So there are clear differences. Why they occur is an open question. One of the questions we're about to start asking with the methods in our lab is when they fold into different shapes, what does their protein neighborhood look like in the cell? Because presumably the way you get a different disease is you muck up different things in the cell, but how do you find them? Some of those could be therapeutic targets and also lead to clues about how simply a change in shape of a protein can lead to a different disease.
Thank you. So I know we're running out of time, but we've had 3 questions on deep brain stimulation, so we'll end on that perhaps. Who are the best candidates for deep brain stimulation?
That's a great clinical question. So there's some important things to note. Really important one is that if you do not respond to dopamine, you will not respond to deep brain stimulation. That's really important. It's not, only patients who have a response to the drugs that we have will end up being candidates for DBS. I think that is one of the most important take-home points. But fundamentally, a good candidate is a patient who has responded well to dopamine for some time, is clearly classical Parkinson's disease, responds to dopamine, but now the dopamine becomes a two-edged sword, and the dopamine, and it's not causing it. Again, I want to correct this, this misunderstanding. Dopamine does not cause side effects like dyskinesias, but as this disease evolves, when you give patients with more advanced disease dopamine, you get those side effects. It's not causing it, so don't hold off taking dopamine. Take dopamine, but as the brain disease evolves, dopamine is certainly associated with side effects. And a patient who responds to dopamine but is struggling with side effects might be an excellent candidate for DBS. I'm not giving medical advice here. You know, there's a full team of neurologists, neurosurgeons, neuropsychiatry as well that weigh into these complex decisions. But this is a really remarkable treatment. It can lower the need for therapy in patients who respond but who are suffering from adverse effects.
Thank you so much. And thank you so much for that fabulous talk. And thank you to the audience for your wonderful questions.
Thank you so much. I really appreciate it.